In the breakdown of fatty acids, beta oxidation, if we have an odd-numbered fatty acid, the last three carbons are going to produce propionyl CoA rather than producing the acetyl CoA and going into directly into the TCA cycle. So instead we have this propionyl CoA, and this propionyl CoA is the last product of beta oxidation in odd-numbered fatty acids. It's going to undergo a series of reactions to produce succinyl CoA, and the succinyl CoA is going to be fed into the TCA cycle. So we start off with this propionyl CoA. It's got the coenzyme A, the double bound oxygen, and the two other carbons. This propionyl CoA is converted into Methylmalonyl-CoA. So this reaction is catalyzed by the enzyme Propionyl CoA carboxylase. And propionyl CoA carboxylase has two different catalytic sites. So we utilize this cofactor biotin, and biotin transfers the reaction from happening from catalytic site 1 to catalytic site 2 of the propionyl CoA carboxylase enzyme. So this reaction also utilizes ATP and this bicarbonate. And bicarbonate is a combination of H2O and carbon dioxide. So what happens is that we have this bicarbonate compound and we have that ATP because we're also utilizing ATP. So the lone pair on this negatively charged oxygen on the bicarbonate nucleophilically attacks that terminal phosphate on the ATP. This causes the bond between the phosphate and that adjacent oxygen to break. So oxygen gains a lone pair and we release ADP. We are releasing ADP because we still have that adenosine and we have the two phosphates, and we only grabbed that one phosphate. So that one phosphate is now bound, so we get this carboxyphosphate intermediate, which is a combination of that bicarbonate and the phosphate from ATP. We released ADP, and this is biotin. Biotin has two catalytic, the pyruvate carboxylase enzyme has two catalytic sites, and what this biotin is going to do, it's going to transfer the reaction from the first catalytic site to the second catalytic site, so C1 to C2. So once we get this carboxyphosphate intermediate, it is a very short-lasting intermediate. This bond between the oxygen and that hydrogen is going to break. These electrons are going to be given to form a double bond between the carbon. So there's a double bond forming here and a double bond forming here. So carbon has to lose a bond because it cannot have 5 bonds. So this oxygen gains a lone pair. So when it gains a lone pair, we have formed inorganic phosphate. So that inorganic phosphate is going to be released. And now when we form that double bond between the carbon, because this hydrogen is lost, we form carbon dioxide, we form CO2. So both these oxygens are highly electronegative, so this induces a very small positive charge on this center carbon. So this center carbon, since it has that very small positive charge, it is able to be nucleophilically attacked by the nitrogen on this biotin. So biotin has this nitrogen with a lone pair, and it will nucleophilically attack that carbon. This causes this oxygen carbon, one of the bonds, to break, and oxygen will gain a lone pair. All these initial steps right now are happening in the catalytic site 1 of the enzyme Propionyl CoA carboxylase. So once that biotin binds with the carbon dioxide, we get this intermediate where this oxygen gained that lone pair. We see this bond forming between the carbon and the nitrogen. So now in the next step is this biotin is going to move this part to catalytic site number 2. So we see this carbon dioxide over here, but now it is over here because we are moving from the C1 to the C2. So we are now in the C2 catalytic site. Now, once we are in the C2 catalytic site with that carbon dioxide, this oxygen with that lone pair, it's highly unstable. It wants to reform that bond with its adjacent carbon. So this lone pair on the oxygen is going to drop back down, form that bond again with the carbon, and this will be released as carbon dioxide once again. But these electrons right over here that formed a bond between the nitrogen and the carbon are given back to the nitrogen, and nitrogen wants to form that third bond, so it's going to form it with that adjacent carbon up here. But this carbon can only have 4 bonds, so it is going to lose one of its bonds to oxygen, and oxygen is going to gain a lone pair. So what happened is that this bond is highly unstable. It wants to reform that bond with carbon. This is going to be released as carbon dioxide. These electrons over here are passed over here to make a double bond between the nitrogen and the carbon, and this carbon, it can only have 4 bonds, so one of these bonds is going to be broken and oxygen gains a lone pair. So when this double bond has formed and this oxygen has that lone pair, oxygen wants to form that second bond. It's highly with the extra electrons that it has, so it's going to reform that bond. These electrons over here are going to abstract a hydrogen off of the propionyl CoA. So this is where we finally see the propionyl CoA added into the mechanism. It is when the nitrogen has formed that double bond and that oxygen has gained a lone pair, and we've released this carbon dioxide in the previous step. So these electrons abstract that hydrogen. This causes these electrons to drop down over here to form a double bond between these 2 carbons. So now that biotin is reformed. So this is a catalytic process. So all these enzymes and cofactors that are being utilized must be regenerated. So the biotin has been regenerated. And over here, we form this enol intermediate where we see the carbon forming a double bond with that carbon because one of the hydrogens over here was abstracted. And this one is missing a hydrogen, so it has a hydrogen as well. So now, what's going to happen is that this enol intermediate that formed that double bond with the adjacent carbon is these electrons are going to attack that carbon because this carbon dioxide has these 2 oxygens which are highly electronegative, so there's a small positive charge on that carbon. So these electrons nucleophilically attack that carbon, and this carbon dioxide group is given to this second carbon over here. And right over here, it should be H3. So the second carbon has gained the COO minus. It's COO minus because this last oxygen, it has a negative charge because once this carbon was attacked, the electrons over here went to the oxygen, so oxygen gained a negative charge, and we see that negative charge right over here. So this is Methylmalonyl-CoA, where we utilized the enzyme Propionyl CoA carboxylase. We used biotin, we used ATP, and we used bicarbonate. With that mechanism, we produced Methylmalonyl-CoA. Methylmalonyl-CoA gained this carboxylate group on the second carbon. So now this Methylmalonyl-CoA is first, the Methylmalonyl-CoA is converted into L-methylmalonyl-CoA. This is done so with the enzyme Methylmalonyl-CoA epimerase. So what's happening is that the coenzyme A that was over here initially is passed on to over here. And we saw in D-methylmalonyl-CoA that there was this COO minus group, which looks like this. So what happened is that the coenzyme A that was over here is given to this oxygen over here. When that's done, this carbon, it loses this coenzyme A, but it's left with an oxygen. So right over here, the that first carbon, it has that oxygen, it lost the coenzyme A. The coenzyme A is passed right onto here, and this is L-methylmalonyl-CoA. Now in the last step, when we're going from L-methylmalonyl-CoA to succinyl-CoA, there's a switch of two functional groups. So this functional group is going to go over here, and this hydrogen is going to be moved over here. And right now in this state, these bonds cannot be easily moved. So there must be some sort of mechanism in place to move these bonds, and that is done so with the help of coenzyme B12. Coenzyme B12 acts as an intermediate to facilitate the exchange of two functional groups. So what happens is that we have this B12, and this B12 is a very large molecule, but we focus on the part that we have this deoxyadenosine bound to the CH2, which is bound to a cobalt at the very middle of B12. So what happens is that this B12, right over here, it undergoes a homeolytic cleavage. And in the homeolytic cleavage, one electron is given to the deoxyadenosine CH2, and the other one is gained by the cobalt. So the cobalt had a +3 charge over here, but once it gained that electron, it now has a +2 charge. Now when we add in the substrate, this radicalized deoxyadenosine is going to abstract the hydrogen off of the substrate. So it's going to abstract that hydrogen. So it gained that hydrogen, and our substrate is left in its radical form. So this, once again, was a homeolytic cleavage. So that substrate is now radicalized, and what's going to happen is this functional group is going to move on to that radicalized carbon. So once that functional group has switched over, the deoxyadenosine that abstracted the hydrogen originally is going to give back its hydrogen. So we get our product. So we saw the switching of the two functional groups. And this is exactly what's happening when we're going from L-methylmalonyl-CoA to succinyl-CoA. We have that hydrogen, and we have that COO with the coenzyme A. The COO coenzyme A moves over, and the hydrogen is donated back, and we get our product. And then we are left with deoxyadenosine, and the deoxyadenosine, in its radical state, they're going to reform that bond, and we're going to get that original structure again, which is ready again to undergo another cycle. So this B12 is acting as an intermediate to facilitate the exchange of two functional groups, and that's done so by radicalizing. So B12 essentially radicalizes the substrate, allows the functional group to switch over, and then the hydrogen is given back, and we get our product. And that is how L-methylmalonyl-CoA is converted into succinyl-CoA. And then this is the structure of the coenzyme B12. We see the CH2 bound to that cobalt, and we see this deoxyadenosine structure over here. So this specifically is the part that is taking part in this reaction. So what happens to the succinyl-CoA? The succinyl-CoA that we produced is going to enter the TCA cycle right over here. It is going to go further into the TCA cycle, and as it does so, it's going to produce this ATP, it's going to produce FADH2, it's going to produce NADH. But the problem is that if we're adding in succinyl-CoA to a catalytic cycle, all these intermediates within the cycle have to have their concentrations kept constant. If the concentrations are not kept constant, this cycle cannot work. So if we're adding in succinyl-CoA, we're increasing that concentration of an intermediate within the TCA cycle. This cycle will not be able to occur. So how do we get rid of that excess of succinyl-CoA? Well, it goes through the TCA cycle. But once we get to either malate or oxaloacetate, we can use an anaplerotic reaction to convert either malate or oxaloacetate into pyruvate or we could convert it into phosphoenol pyruvate, and phosphoenol pyruvate is the second last intermediate of glycolysis. So right before pyruvate, we would have phosphoenol pyruvate. So the excess succinyl CoA, it goes further into the TCA cycle. When it gets to either NADH or oxaloacetate, it can be converted into pyruvate, and that pyruvate can be converted into acetyl-CoA, and that acetyl-CoA can go through that cycle and produce energy. So that is how we get rid of excess. We use anaplerotic reactions to convert either malate or oxaloacetate into pyruvate. The excess has been removed, and that pyruvate is further converted into acetyl-CoA and then further gone into the TCA. And these are the 4 common anaplerotic reactions. So if we have oxaloacetate, it can be converted into pyruvate, or it can be converted into phosphoenol pyruvate, or there's this reaction, or if we have malate, it can be converted into pyruvate. So that is how the propionyl CoA that is produced by beta oxidation, when we have an odd-numbered fatty acid, we get propionyl CoA. It's a 3-carbon molecule. It is broken down into D-methylmalonyl-CoA, then it becomes L-methylmalonyl-CoA, then to succinyl-CoA. Succinyl-CoA enters the TCA cycle. It goes through the TCA cycle, and then in order to remove the excess, we use these anaplerotic reactions to convert that excess, either oxaloacetate or malate, into pyruvate or phosphoenol pyruvate, and then they can be converted into acetyl-CoA and then further undergo the TCA cycle and produce energy.